This protocol begins by adding incubation buffer, Fitzy casein solution, and sample containing the protease of interest to a microcentrifuge tube, and then gently mixing and incubating at 37 degrees Celsius for 60 minutes in the dark. To stop the proteolytic reaction at the end of the incubation period, add TCA solution, gently mix, and incubate at 37 degrees Celsius for 30 minutes, again in the dark. This step will cause the unreacted substrate to precipitate. Centrifuge the sample for 10 minutes at 10,000 Gs. Remove the supernatant, which contains the acid-soluble Fitzy labeled fragments. Transfer the supernatant to a cuvette or multiple well plate, dilute with assay buffer, and read the sample on a fluorimeter.
Hi, I'm Carrie Cup-Inyard, and I'm a scientist at our Sigma Aldrich DeKalb facility in St. Louis, Missouri. Today I will be showing you a procedure for fluorometric protease activity determination. We use this procedure in our quality control lab to determine low protease activity. So let's get started.
For this procedure, the incubation buffer, assay buffer, and 0.6 normal trichloroacetic acid, or TCA solution, are provided as ready-to-use solutions. Aliquot the solution of Fitzy-labeled casein into smaller volumes and store at -20 degrees Celsius, making sure to protect from light by using amber vials or wrapping clear tubes in foil. Aliquoting the Fitzy casein substrate minimizes the need for repeated freeze-thaw cycles. Repeated freeze-thaw cycles, or vigorous shaking, or mixing of the Fitzy casein could lead to separation of the Fitzy from the casein, resulting in higher background. If calibrating the fluorimeter or determining the linear range for the Fitzy signal, prepare the Fitzy control solution. Make the solution fresh by reconstituting in assay buffer to the appropriate concentration. Once ready, protect the solution from light. Finally, prepare the trypsin protease control by adding 100 microliters of 1 millimolar hydrochloric acid to the vial of lyophilized trypsin, and mix briefly to ensure the trypsin is dissolved. Storing the trypsin under acidic conditions increases its stability. Note that the trypsin control solution is temperature sensitive and is not stable at low concentrations. Next, start preparing all of the assay samples, which include test samples, trypsin control, and blank. Throughout the assay, keep the samples protected from light. For each test sample, combine in a microcentrifuge tube 20 microliters of incubation buffer, 20 microliters of the Fitzy casein substrate, and 10 microliters of the test sample. One may use a clear microcentrifuge as long as the sample is incubated in the dark. Note also that test samples which have high protease activity will need to be diluted. The trypsin control sample is used as a positive control that the assay is performing properly, to determine the detection limit, or to create a general standard curve. To prepare the trypsin control, combine 1 part acidified trypsin to 9 parts of incubation buffer, or the buffer used for the test samples, and mix well. The final working concentration of trypsin is 20 micrograms per milliliter. If using a different specific protease, prepare the control solution with that specific protease and an appropriate buffer. Continue to prepare the control sample by combining 20 microliters of incubation buffer, 20 microliters of Fitzy casein substrate, and 10 microliters of the trypsin control in a microcentrifuge tube. It is recommended to run at least one control sample of 5 nanograms trypsin with each assay, or to use serial dilution. The last sample is the blank, prepared by combining 20 microliters of incubation buffer, 20 microliters of the Fitzy casein substrate, and 10 microliters of ultra-pure water in a microcentrifuge tube. With the 3 types of samples ready, gently mix each tube and incubate at 37 degrees Celsius in the dark for 60 minutes. Be careful not to mix too vigorously, as excessive turbulence may cause high fluorescence background and reduce the sensitivity of the assay. One can incubate up to 24 hours to increase sensitivity, but longer incubations are not recommended, as the FITC casein may begin to degrade, again leading to high fluorescence background. When the incubation is complete, use appropriate protective equipment to add 150 microliters of the TCA solution to each microcentrifuge tube. Upon incubation with TCA, one should observe a white haze present in the tube, which is the precipitated, undigested, and insoluble casein. Mix gently and incubate at 37 degrees Celsius in the dark for 30 minutes. At the end of the incubation with the TCA solution, centrifuge the tubes for 10 minutes at 10,000 Gs at room temperature. The supernatant contains the acid-soluble, FITC-labeled casein fragments used for the fluorescence measurement. Pipette gently from the top of the tube so that none of the precipitate in the pellet is drawn into the pipette tip, which will cause a false positive readout during the following fluorescent detection. To measure the protease activity using a fluorimeter, combine 10 microliters of the TCA supernatant with 1 milliliter of the assay buffer in a tube and mix gently. In addition, dilute the FITC standard for analysis by adding 10 microliters of FITC to 1 milliliter of assay buffer. The FITC control and trypsin standard give a fluorescence level that the sample should not exceed. The solution of the TCA supernatant and assay buffer may be stored in the dark at 2 to 8 degrees Celsius for up to 24 hours before measuring the fluorescence. If measuring using a cuvette, transfer 1 milliliter of the solution to a suitable cuvette. If a large number of samples are to be run, a 96-well plate and fluorescent plate reader may be used to acquire fluorometric data. In this assay, fluorescein isothiocyanate is provided as a control for possible instrument calibration. Shown here is a representative linearity range of the FITC signal. Following the described procedure using this kit, with an incubation time of 30 minutes for the first incubation step, yields this typical standard curve for trypsin and control samples, ranging from 1.5 nanograms to 25 nanograms. Fluorescence measurements were made in a multi-well plate. The limit of detection of the assay is the amount of protease that produces a significant fluorescence reading above the value obtained with the blank sample.
Today, we've shown you how to use the Sigma-Aldrich Protease Determination Kit to detect fluorometric protease activity in a biological sample. While this kit is optimized for your use, it's important to remember that for your particular proteases, you may need slight modifications. So that's it. Thanks for watching, and good luck with your experiments.